Hi everyone, I am Dr. C.S. Kulkarni, MD, and I am going to teach medical biochemistry. Before starting the topic, please like, share, and subscribe the channel. In the last session, we studied about peptide bond and primary structure of proteins. In today's session, we will further study primary structure of proteins. Proinsulin. Beta cells of islet of pancreas synthesize the insulin as a prohormone. Beta, sorry, proinsulin is a single polypeptide chain with about 86 amino acids. Biologically active insulin that has two chains is formed by removal of the central portion of the proinsulin before release. The C peptide, that is the connecting peptide, is also released into the circulation. In this diagram, we can see that proinsulin breaks into insulin and a connecting peptide. In the primary structure of in the primary structure determines biological activity. A protein with a specific primary structure, when put into a solution, will automatically form its three-dimensional shape. The higher levels of organization are dependent on the primary structure. In this diagram, we can see that tertiary protein contains structure is dependent on primary protein structure. Even a single amino acid change, that is mutation, in the linear sequence may have profound biological effects on the function. For example, the hemoglobin A, which is a normal hemoglobin, the sixth, the sixth amino acid in the beta chain is glutamic acid. It is changed to valine in hemoglobin S, that is sickle cell anemia. In this diagram, we can see that HBS is formed from HPA due to the change in sixth amino acid of beta, that is from glutamic acid to valine. Secondary structure of proteins. The term secondary structure denotes the configurational relationship between residues which are about 3 or 4 amino acids apart in the linear sequence. Secondary and tertiary levels of proteins. Structure are preserved by non-covalent forces or bonds are like hydrogen bonds, electrostatic bonds, hydrophobic interactions, and van der Waals forces. In this diagram, the hydrogen bond is a weak electrostatic attraction between one negative electronegative atom like oxygen or nitrogen and a hydrogen atom covalently linked to a second electronegative atom. In this diagram, we can see that a weak hydrogen bond is formed by an oxygen of one water molecule and a hydrogen from another water molecule. Electrostatic bonds or ionic bonds. Positive charges are donated by epsilon amino group of lysine or quinidinium group of arginine and imidazolium group of histidine. Negative charges are provided by beta and gamma carboxyl groups of aspartic and glutamic acid. In this diagram, we can see that at one atom shares another electron with another atom to form ionic bond. The hydrophobic bonds are formed by interactions between non-polar hydrophobic side chains by eliminating water molecules. This serves to hold lipophilic side chains together. The van der Waals forces are weak but collectively contribute maximum towards the stability of the protein structure. In this diagram, we can see that two hydrophobic or lipophilic groups come together in space by hydrophobic interactions. Alpha helix. Pauling, who was awarded Nobel Prize in 1954, and Corey described the alpha helix and beta plated sheet structures of polypeptide chains in 1951. The alpha helix is the most common and stable conformation for a polypeptide chain. In proteins like hemoglobin and myoglobin, the alpha helix is abundant, whereas it is virtually absent in chymotrypsin. The alpha helix is a spiral structure. The polypeptide chains form the backbone, whereas the side chains of the amino acid extend outwards. The structure of stabilized hydrogen bonds between NH and CO groups of the main chain. In this diagram, we can see the alpha helix, which has its center formed by polypeptide chains, whereas side chains extend outwards. Each turn has formed is formed by 3.6 residues. The distance between each amino acid residue, that is translation, is 1.5 angstrom units. The alpha helix is generally right-handed. Left-handed alpha helix is rare because amino acids found in the protein are of alpha variety, which exclude left-handedness. Proline and hydroxyproline will not allow the formation of alpha helix. Beta pleated sheet. In the polypeptide chains in beta pleated sheet is almost fully extended. The distance between adjacent amino acids is 3.5 angstrom units. It is stabilized by hydrogen bonds between NH and CO groups of neighboring polypeptide chain segments. In this diagram, we can see that there is a structure of beta pleated sheets which are held by hydrogen bond. Coming to the difference between alpha helix and beta pleated sheets. In alpha helix, right-handed coiled rod-like structure is present, whereas beta sheets, a sheet-like structure is present. In alpha helix, hydrogen bonds form within the polypeptide chain, whereas in beta sheets, there are two form I linking two or more beta strands by hydrogen bonds. In alpha helix, hydrogen bonds are formed between NH group of one amino residue with CO group of another amino acid, whereas in beta sheets, hydrogen bonds are formed between the neighboring NH and CO groups of adjacent peptide chains. In the alpha helix, the R groups are the amino acids are oriented outside the helix, whereas R groups directed to both the inside and outside the sheet. Alpha helix can be a single chain, whereas beta sheet cannot exist as a single beta strand. Alpha helix has only one type, whereas beta sheets can be parallel, antiparallel, or mixed. Alpha helix prefers alanine, leucine, metformin, phenylalanine, glutamic acid, glutamine, histidine, lysine, arginine, and amino acids, whereas beta sheet prefers tyrosine, tryptophan, phenylalanine, metformin, isoleucine, valine, threonine, and cysteine. Adjacent strands in a sheet can run in the same direction with regard to the amino and carboxyl terminal ends of the polypeptide chain, that is parallel, or in opposite chain, that is antiparallel beta sheet. Beta period sheets is the major structural motif in proteins like silk fibroin that has antiparallel, then flavodoxin that has parallel, uh, carbonic anhydrase that has both. In this diagram, we can see that parallel and antiparallel beta sheets. In this diagram, beta bends may be formed in many proteins by abrupt U-turn folding of the chain. Intrachain disulfide bridges stabilize these bends. Collagen helix. It is the triple helical structure found in collagen. In this diagram, we can see the triple stranded collagen structure. Tertiary structure. Secondary structure denotes the configurational relationship between residues which are about 3 to 4 amino acids apart, or secondary level defines the organization of immediate vicinity of amino acids. The tertiary structure denotes three-dimensional structure of the whole protein. The tertiary structure defines the steric relationship of amino acids which are far apart from each other in the linear sequence but are close in the three-dimensional aspect. Tertiary structure. The tertiary structure is maintained by non-covalent interactions such as hydrophobic bonds, electrostatic bonds, and van der Waals forces. The tertiary structure acquired by the protein structure is always thermodynamically stable. Domain is the term used to denote a compact globular functional unit of a protein. A domain is a relatively independent region of the protein and may represent a functional unit. The domains are usually connected with the relatively flexible areas of protein. In this diagram, we can see that the repeat structure like WD-40 repeat domain, leucine rich domain, and so on. Phenylalanine hydroxylase enzyme contains three domains. One regulatory, one is catalytic, and one protein-protein rich interaction domains. Quaternary structure. Certain polypeptides will aggregate to form one functional protein. This is referred to as quaternary structure. The protein will lose its function when the subunits are dissociated. The forces that keep the quaternary structure are hydrogen bonds, electrostatic bonds, hydrophobic bonds, and van der Waals forces. Depending on the number of polypeptide chains, a protein may be termed as monomer that has one chain, dimer that has two chains, tetramer that has four chains, and so on. Each polypeptide chain is termed as subunit or monomer. Homodimer contains two copies of the same polypeptide chain, whereas heterodimer contains two different types of polypeptides as function units. For example, two alpha chains and two beta chains form the hemoglobin molecule. Similarly, two heavy chains and two light chains form one molecule of immunoglobulin G. Creatine kinase is a dimer, whereas lactate dehydrogenase is a tetramer. Here ends our session. Please like, share, and subscribe the channel. Thank you.